And now it's my great pleasure to welcome Juliette Legler as our speaker of today's Eawag seminar. Juliette is a professor of toxicology and heads the Division of Toxicology at the Institute of Risk Assessment Sciences at the Utrecht University in the Netherlands. And since 2020, she is also the head of a large department called Population Health Sciences. And before that, Juliette was a professor of toxicology and environmental health at the Brunel University London, and she is still affiliated there as an honorary professor. From 2001 to 2015, she worked at the Institute of Environmental Studies at the Vrije Universiteit Amsterdam, where she was appointed professor of toxicology and environmental health in 2013. So Juliette and her group have done pioneering work on endocrine disruptive chemicals, so chemicals that interact with the hormone system, and with special focus on the developmental exposure of these chemicals. And actually work from her group provided evidence for a link between the developmental exposure of endocrine disruptors and the vulnerability to develop metabolic diseases such as diabetes and obesity. And Juliette has also won several awards for this work and has coordinated a large EU-funded project on this. She's now also the coordinator of a large consortium that develops test methods to identify metabolism disrupting chemicals. And more recently, she also attracted funding to do pioneering work on microplastics in human fetal environment. And although more focusing on human health now, her research has also always been involving animal and environmental health. And she is strongly promoting the concept of One Health, saying that everything is interconnected. And to have healthy human beings, you actually need healthy animals and a healthy environment. So this might seem quite obvious to some of you, but unfortunately, human health sciences and environmental health sciences often appear to be largely separated fields. And Juliette manages very well to bridge between these two fields. And today, Juliette is going to present her newest findings on the risks of microplastics to human health. And 2 weeks ago in the Eawag seminar, we actually learned that people are perceiving the presence of microplastics as a risk to human health, but the real impact is actually largely debated. So Juliette today will give us some insights on the evidence and knowledge gaps in this topic. And so this is actually the perfect follow-up seminar on our microplastics seminar 2 weeks ago. So Juliette, thank you very much for preparing this talk for us today. And I'm now handing over the word to you.
Thank you very much, Colette. Can you hear me okay?
Yeah, we can hear you very well.
Very good. Thank you very much for this very, very nice introduction. It's always very nice to hear that work. And indeed, I've been working in the field of endocrine disrupting chemicals for a long time and was very much interested in this field of microplastics, realizing how little we actually know about potential health risks to humans. So it's a new field for me, and I've been learning a lot and learning, of course, using my knowledge from the endocrine disruptor field in this field as well. So it's a pleasure for me to talk to you today about this new field of microplastics and human health. And I hope that I can show you some new work, the design of a new project that will start, and also convince you that there are really key knowledge gaps in this field that need to be filled before we can say anything about the risks of microplastics to human health. So Colette mentioned already that I look at research from a One Health perspective. It's interesting. I've been working at the Faculty of Veterinary Medicine for the last 3 years, and I wanted to use this One Health approach to really look at microplastics. So I have this this meeting is being recorded. Continue. There it's gone. Hopefully you can see my slides now. Before I get into a One Health approach to microplastics, I wanted just to briefly introduce what these microplastics are, in case you're not familiar with them. When we are talking about micro and nanoplastics, we're talking about particles made from plastic that have emerged from the weathering or breakdown of plastics, and they can be become very, very small in size, and microplastics being less than 5 millimeters in diameter, and nanoplastics really less than a millimeter. And generally, the plastics, a micrometer, the plastics about 100 nanometers or 200 nanometers or smaller are considered the nanoplastics. This slide, which I've taken from a publication, a recent publication, shows very clearly that plastics are a complex, microplastics are a complex mixture of many different factors. So on the left, you'll see that plastics, of course, as we know, are many different kinds of plastics, of which these microplastics could be made of. The plastics in themselves are polymers that also contain many additives. So many different chemicals are used to provide plastics with their particular qualities that we use them for. Microplastics can be made from primary products, so they can be actually made intentionally for different applications, but they are generally secondary products arising from, as I mentioned, the weathering or breakdown of many different larger plastics used in all sorts of different applications. So I mentioned the size already. So not only is the size really important in the potential health risk of the microplastics, also the morphology, so the shape of the plastic particle is important. So they can be very diverse in shape, from round plastic spheres, where most of the research up till now has taken place, to much more complex particles that can be fibers or fragments or films or foams. In addition, these plastics can be different colors. As you can see on the picture on the bottom right hand side of the slide, they are very diverse in shape, size, and color, chemical composition, and they may also absorb different chemicals from the environment, such as these so-called ecotoxins shown here on this slide. So a very diverse mixture of particles. Here's to give you an indication, just in the country that I live in the Netherlands, how many tons per year of emissions are being emitted from of microplastics from different sources, from litter fragmentation, so plastic fragmentation of plastics on land, to tires. The wear and tear of tires is a large source of microplastics, from paint products, from pellets, from cosmetics, from washing our synthetic clothes in the washing machine can lead to these microplastic fibers being formed, cleaning agents, etc. So there's a diverse sources of these plastics, and the numbers of production in terms of tons are very large. So should we be concerned? Well, let's look at microplastics from a One Health perspective. And coming from a veterinary faculty, One Health being really important in particular in understanding the role of disease between humans and animals, but can also be translated to the role of the environment. And as Colette said in her introduction, the idea being of this One Health paradigm, that the health of our environments, of our animals and our people are really interrelated, really dependent on each other. And we cannot really look at the health of one of these factors in isolation. They all play a role in each other. So when we look at microplastics, the microplastics field originated more from the concern of impacts of plastics in our environment. So focusing in the first place on the idea of microplastics in our environment and their particular role on the effects on the health of an environment, we know that that plastics in the environment are of concern. If we look at the plastics that are being emitted into our oceans, we know that a lot of plastics have been end up in our oceans. As you can read here, 80% of microplastics coming from land entering rivers are delivered ultimately to our oceans. Oceans being filled with staggering tons of plastic. even seems some calculations predicting that there is more plastic in our oceans in terms of tons of plastic than there are tons of fish. I think that's always a very striking comparison for me. So plastics in our oceans being broken down into these microplastics is as of concern in terms of the actual long-term persistence of these contaminants, this pollution in our aquatic systems, and the ultimate effect on organisms in the aquatic environment. Recent research is showing that we should not only be concerned with the huge amount of plastics in our oceans and the related numbers of tons of microplastics, but also microplastics in our air are of concern. So the plastic cycle is shown in this slide here, indicating that what is discharged in our terrestrial environment will end up in our oceans, but will also be cycling into the atmosphere, with recent studies indicating that a significant portion of atmospheric pollution is made up of plastics and of microplastics. So potentially microplastic exposure to humans and to animals is not only through exposure in the water, but also through inhalation and exposure through microplastics in our air. So this is a nutshell indicating that there's an environmental concern for the presence of microplastics in both aquatic environments, as in terms of the atmospheric exposure to microplastic pollution. So what about the impact then on animals, on organisms in the environment? So considering the potentially very, very high exposure to microplastics through water and through air, most studies in looking at health risks of microplastics have focused indeed on organisms in the environment, and not so much on humans, because these organisms could be directly and are directly exposed to microplastics. Now, this overview slide gives us an idea of how microplastics could actually accumulate in our food chain and in aquatic food chains of aquatic organisms, indicating that, and also studies showing that microplastics can be uptaken by lower organisms at lower levels of the food chain, where they can be transferred to higher levels of organization. And so microplastics have been found in many different species in aquatic environments, leading right up to higher levels of biological organization and top predators, including seabirds and marine mammals. So that's an example of we knowing that there is exposure to animals in the environment, knowing that these microplastics can reach most pristine corners of our Earth due to atmospheric and transport through water. The question being, what are the actual potential effects of this form of pollution? Well, microplastics and nanoplastics can provide an interesting study for the toxicologist. Certainly for myself, focusing on that chemical effects of chemicals, endocrine disrupting chemicals that can be leached from plastics, I realized when studying microplastics that it's a lot more complex. A plastic particle is made up of different components, as I introduced earlier, that could all have their own potential toxicity. So a microplastic is, as I mentioned, a particle shown here on the left that could be of different sizes and shapes. A particle in itself can cause toxicity when it enters a tissue or a cell. Generally, this type of toxicity is toxicity that relates to a stress response in the cell, to a reaction of the cell to try to fight against the presence of this particle, for example, by an immune reaction, by the recruitment of different immune cells to try to clean up and remove the particle from the cell. So particle toxicity encompasses first effects of the actual particle entering the biological system and include oxidative stress responses, membrane toxicity, but also immune reactions in the tissue and in the cells. So the microplastic can cause particle toxicity. In addition, the microplastic could potentially cause chemical effects in that, as I mentioned earlier, the particle contains additives, chemical additives that have been added to the plastic to give the plastic its specific shape and characteristic or form or softness or hardness. Right? Lots of additives are added to plastic. These chemicals could leach out into the biological system where they could cause toxicity. Studies up till now are indicating that perhaps this is maybe not the major source of toxicity of a chemical, of a plastic, microplastic. However, what we haven't really studied yet is the hypothesis that through transport through our entire body, these microplastics, or through the body of an organism, these microplastics might reach specific cells and tissues where they leach out these plastics and might cause really in situ specific effects in those specific cells. It's an area that has not been studied much yet. So in addition to the particle toxicity and the chemical effects, there is also concern that microplastics may have so-called microbial effects or pathogenic effects. And this has to do with the phenomenon that these particles, these microplastic particles, are covered with a film of microorganisms, proteins and other molecules that can stick to the outside of the plastic particle and could serve as carriers for pathogenic, types of bacteria or viruses that may cause sickness or adverse health. So the third form of toxicity being a potential vector for diseases in terms of the particle transporting and being a vector of these bacteria and other dangerous microorganisms that may stick to the outside of the plastic particle. So I'll come back to these different forms of toxicity as I move forward in this presentation. So if we look at the studies that have been done in the field using organisms in the environment, using different laboratory studies with model organisms, such as fish, such as invertebrates, such as crustaceans, various species that have been tested to look at effects of microplastics on animals, organisms in the environment, adverse effects. There have been many studies showing adverse effects in over 100 species in laboratory experiments. In general, these studies may have their caveats. In general, exposure concentrations in the laboratory tend to be high in order to look at the potential hazard of these microplastics to the organisms studied. So because there's not much known about the actual exposure concentrations in the field, the studies will focus on higher exposures to see at what levels effects can be found. And this may not be realistic exposure concentrations. We need to keep that in mind. It's also clear that the laboratory studies with micro and nanoplastics are done often with particles that are of a different size or plastic type than those that are reported in the field. So in general, the field is quite new. There have been many studies done with commercially available microplastics of a specific size and shape, whereas we know in the fields this will be much more complex and many more sizes will be available for exposure to the organism. And up till now, limited studies have been done to really look at effects of microplastics in the field. So the figure on the bottom of this slide gives an indication of the types of effects that have been found in these laboratory experiments with different species. As I mentioned, fish, invertebrates, crustacean species. Generally, a lot of work has been done on the aquatic organisms because of the high exposure in aquatic environments. So you can see here different molecular and cellular effects, different systemic effects, and different effects on individual organisms that have been reported for exposure to microplastics. So a lot of evidence coming from animal studies in laboratories and limited evidence in the field, but certainly evidence that's indicating that microplastics are taken up by organisms where they may cause effect. So the question is, what are the effects on humans? And I must say this is still a really young field. However, I personally would say that if a microplastic is taken up by a fish or by another vertebrate organism, I have really no reason to believe that it wouldn't be taken up by a human. However, the field is definitely in its infancy, and we need to do some really critical research to understand better the risks to people. So together with a colleague of mine, Professor Fethak, we recently published a paper just about a month ago in which we tried to summarize the major knowledge gaps that are really needed to address the human health effects of microplastics. In this paper, we've summarized for the reader, the main sources of plastics, of microplastics. We have a number of reliable and good external exposure measurements showing that humans can be exposed through inhalation or through diet to microplastics coming from various sources, as you would see in the top left-hand side of this figure. Again, we emphasize the importance of the complex and diverse characteristics of these microplastics, acknowledging that most of the research up till now has been done with plastic beads, where we really know very little about other shapes and compositions. We indicate that the possible human health effects that we have seen from the publications up till now indicate that there may be DNA damage, may be cellular damage, and may be immune-related effects related to inflammation, but really it's really just the tip of the iceberg, and a lot more research is necessary to better understand potential human health effects. So what are the main knowledge gaps that we have in terms of human health risks of microplastic exposure? So the number one knowledge gap, in our opinion, is the gap of exposure data. So as I mentioned earlier, there is considerable exposure data when it comes to understanding what humans are exposed to externally. So what is potentially in the air or in our diet. So what could be taken up through lungs or ingestion. Though I should mention that this research is focused mainly on the larger sizes of the microplastics and not so much on the nanoplastics. And this has everything to do with the analytical challenges with measuring these kinds of tiny, tiny particles. Whereas on the one hand, external exposure, large strides are being made to improve exposure assessment. Externally, we know little to nothing about the actual internal exposure of humans. There is a really urgent need for analysis of human tissues, for understanding what is actually taken up in, for example, human blood, to better understand systemic exposure in humans, but also to understand where the specific tissue uptake and accumulation of plastics could occur, to better understand if plastics are in critical tissues such as the placenta or the brain. I'll come back to that in a moment. So really understanding this exposure internally of small micro and nanoplastics is a huge data gap that needs to be filled in order to understand the risk. There are some other data gaps when you look forward towards toxicity, understanding toxicity. First of all, what is the uptake of these complex mixtures of micro and nanoplastics uptake over various membranes into various cell types. The first studies indicate that there's potentially a low uptake over membranes of the plastics that have been studied up till now. However, we do not know what long-term exposure to micro and nanoplastics means, because we haven't done those kinds of studies yet. We don't understand how important size is in the uptake, size and shape, I should write here, over human epithelial membranes and barriers. We don't understand how the external, the film surrounding the microplastic, which we call the corona. In this case, I'm not referring to the corona, the virus, but the actual name for the eco or biological film surrounding the plastic, which will be formed during the plastic being in the environment, but also as the plastic is taken up in the body, it will undergo changes and there will be differences to this external coating of the plastic, which we call the biocorona. What does that do to the uptake and toxicity of the plastic particle? We know very little about the absorption, distribution, and metabolism and excretion of these micro and nanoplastics. When it comes to toxicity, again, preliminary studies are indicating specifically particle toxicity, so immune reactions to the micro nanoplastics that require further study to understand. Chemical toxicity seems to be less of a problem than particle toxicity. However, again, more research is necessary there. And really very, very little is known about the potential toxicity caused by the uptake of pathogens that are associated with that corona surrounding the plastic particle. So ultimately, it's really these types of gaps need to be filled before we can say anything about risk, before we can understand the potential risk and understand which plastics we really need to focus on in order to take intervention and mitigating steps. What we can really do is understand that plastics may have a lot of, these microplastics may have a lot of parallels with what we already know from air pollution studies. We already know that air pollution is one of the most important environmental sources of disease and mortality globally. We know that microplastics make up air pollution. So there is a significant source of evidence coming there for the importance of microplastics for the environmental burden of disease due to these parallels with air pollution. We also have learned a lot when it comes to risk assessment of nanoparticles used in the field of nanotechnology and nanomedicine. I think we can learn a lot from those fields and would help us to move our risk assessment frameworks forward. So let me look at the time. It's about 4:30. So in the last 10 minutes of my presentation, I want to give you some evidence of our first, particularly in my lab, our first studies to try to answer some of these questions. So in the Netherlands, I think the Netherlands has been a pioneer in the world when it comes to funding research initiatives that really focus on human health, or potential human health effects of exposure to microplastics. So last year, actually in 2019, the major funding agency in the Netherlands for medical research, that's the Netherlands Organization for Health Research, has funded research into human health effects of micro nanoplastics. They funded a series of 15 so-called breakthrough projects, and these were projects of 1 year long, in which they were quite high-risk projects. They were meant to answer some of the most pressing questions regarding human health, potential human health effects related with microplastics. So of those 15 projects, my laboratory was granted 3 different projects. As they you can see the titles of the projects here. Various researchers in my group worked on these questions. We looked at how microplastics are taken up across the intestinal barrier, and if they would cause immune reactions in the gut. We also looked at how microplastics could affect the brain, in looking at how microplastics affect neurons, the function of neurons, and looking at an in vitro model. So we looked at how we could replicate the blood-brain barrier, this complex cellular barrier in the brain that is there to stop chemicals and certain molecules from passing from the bloodstream into the brain. It's called the blood-brain barrier. We looked at how these microplastics could pass the blood-brain barrier using our own human model of a blood-brain barrier. And finally, we devised a project in which we looked at microplastics in the human fetal environment. So I was in charge of that last project, and that's where I'd like to spend a few minutes talking to you about how we designed that project and some of our preliminary results from that project. I won't go into the other projects because all of these projects are really just completed in December of last year and are in the process of being published. If you have any questions about them, I'll be happy to try to answer them and refer you to the main researchers involved there. But let's take a look at that third project in which we looked at potential effects of micro nanoplastics in the human fetal environment. So where did we start? We started with the hypothesis that micro nanoplastics would end up in the placentas of our pregnant mothers. There was absolutely no evidence when we started this project for that. There had been no studies done to look at the presence of micro nanoplastics in human placentas. However, we had a couple of lines of evidence that made it quite clear that we could expect that micro nanoplastics would be in the human fetal environment. So first of all, the first line of evidence being the fact that air pollution and particles from air pollution can accumulate in human placenta samples. This is some figures taken from the media attention, but also the actual Nature communication paper of Bove and colleagues in 2019 that showed that black carbon, which is these are very, very tiny particles in the nanometer size range, are present in human placenta samples. And these black carbon particles are coming from air pollution and inhaling polluted air of these pregnant mothers. So clearly, if black carbon particles will accumulate in the placenta, there's really no reason to believe that the microplastics of the same size would not. That was our hypothesis. We also know from various studies in animals, for example, studies in pregnant rodents, that if you inject animals with microplastics, in this case with fluorescent microplastics, that the fluorescent microplastics will end up in the embryos and the fetuses of these pregnant animals within hours after injection into the bloodstream. So of course, injection into the bloodstream is not a really biologically relevant exposure route, but it gives us already a worst case scenario in a pregnant organism that these microplastics will enter circulation and will end up in fetal tissues, including the placenta, the brain, the lung, and the liver. Just a few months ago, the first human study was released showing that microplastics are indeed in human placenta samples. So on the left here is some media attention that was given in the Netherlands, but this article that came out in Environmental International at the end of last year was given international media attention, received a lot of international media attention because it's the first study showing micro and nanoplastics in human placenta samples. And what these authors showed was clear indications of uptake of different particles originating from different types of plastics in human placenta. So we have the proof of principle that microplastic particles are taken up in the placenta. When we started the study, they weren't there. We didn't know this yet. We didn't know whether or not we could actually measure micro and nanoplastics in the human placenta. When we started out this study, we posed 3 questions. First of all was, do microplastics reach the human fetal environment? The human fetal environment for us being the placenta, which is of course a really important temporary organ that develops during pregnancy to provide nutrition and oxygen and vital components to the developing baby. We wanted to know whether the microplastics would reach the placenta. We know that chemicals such as endocrine disrupting chemicals that are in the placenta may be transferred to the baby. The placenta is not a non-penetrable organ. It's actually is in exchange with the developing baby, and numerous chemicals and molecules have been shown to be transported from the placenta into the fetus. We wanted to know if micro and nanoplastics would be present in the amniotic fluid, which is the liquid compartment surrounding the fetus. The baby constantly swallows the amniotic fluid during development. So anything that's in the amniotic fluid is in direct contact with the baby and will be directly taken up by the baby. So we wanted to know if they would be taken up through the amniotic fluid and the placenta, and if they would cause harm in the placenta, and if chemicals associated with the microplastics might cause harm to the developing fetus. So the only way, I think, to tackle these kinds of complex questions, and the only way that we can tackle the question, do microplastics cause adverse health effects in humans, is to really work in multidisciplinary teams. So this is what we did for this relatively small project, but this is what we're doing in the future of microplastic and human health research. We're working in teams. We're working with chemists, with pediatricians, with environmental scientists, with toxicologists, with epidemiologists and cell biologists to try to answer these questions. In this particular project, we worked with a pediatrician who provided us placenta samples taken at delivery and provided us with amniotic fluids samples taken when the babies were born from female volunteers here in the Netherlands. We worked with chemists in Amsterdam, chemists in the US, who both of these groups of chemists having their own robust methods or developing their robust methods for measuring micro and nanoplastics in human tissues. Our environmental scientists provided us with environmentally relevant microplastics to study in toxicology experiments. Our epidemiologists advised us on how we could analyze the data coming from the chemistry work. Our cell biologists helping us with imaging and really visualizing the microplastic particles within placenta cells. And we as the toxicologists performing studies in the laboratory to try to better understand the uptake of microplastics in the placenta cells and the biological responses in those cells when they're exposed to these plastic particles. So in order to detect microplastics in human placenta and amniotic fluid samples, we worked on 2 different methods, one in the Netherlands, one in the US. One's based on pyrolysis gas chromatography, mass spectrometry. The other method working on more liquid chromatography, high resolution mass spec method in order to detect different polymer signatures in the placenta and amniotic fluid samples. We measured 30 samples from the Netherlands and 30 samples from the US. These methods, it took a long time to develop the methods because they need to be extremely sensitive, but they also need to be able to distinguish that the plastic signatures are really coming from the actual samples and not from any sort of background contamination from the laboratory or from the sampling of the the samples. I cannot stress enough how important good quality assurance and quality control is in these methods, because plastics are everywhere and the potential background contamination is always present. So the chemists working on the placenta samples are now in the process of publishing their work. So I have to be really, really careful with what I show here. I can tell you that we have strong indications that micro and nanoplastics are present in human placenta samples and in human amniotic fluid samples. I can't show you any data yet until we've published, but those indications are there. When we look at the actual toxicity of the micro and nanoplastics in the human placenta, of course, we don't do experiments on humans. What we do is we use the best model possible in our laboratory, and we try to mimic the situation in the laboratory. What we've done is we've been working with placenta cells that we can culture in a petri dish, and we've been exposing them to various kinds of microplastics to look at the uptake of these microplastics in the placenta cells. And to do this, you need really good microscopes, and that's why the work together with the cell biologists at the biology imaging facility is so important. So this work can give us these kinds of images that, for example, here, this kind of a film that we can make of the placenta cells exposed to, in this case, nanoplastics made of polystyrene. These are round plastic beads. We've also done this work with fibers, where we've also seen uptake of microfibers into the placenta cells. So what I'm going to show you now is a little film. These are placenta cells in a petri dish. The big blue balls indicate the cell nuclei, the yellow indicating the different cells being stained with a different protein. So these are all placenta cells with big blue nuclei. And what you'll see now is in the film is a sort of images taken going from the top to the bottom of the cell and reconstructed to show you where the purple fluorescent microplastics are in these placenta cells. So these purple balls indicate the microplastics that we've added to the cells. And when we take the images of these fluorescent microplastics, we can really clearly see that they are inside the placenta cell. They are not on top, that they have gone through the membrane of the placenta cell, are inside the placenta cell. So this is for us really important to confirm that these cells do indeed take up these very, very small microplastics. In fact, our research indicated that, that we can show that these very small microplastics, right up to 10 micrometer larger plastics, are taken up by placenta cells. Two kinds of different placenta cells that mimic early pregnancy, placenta cells that mimic how the placenta develops as pregnancy moves forward. We can confirm that these cells do take up these microplastics. And when we go on to measure cellular responses inside the cells, we see that at the level of the genes, different genes are expressed differently in cells that are exposed to microplastics than in cells that are not. We also see that these different metabolites are expressed in these cells that indicate that the cells that are exposed to microplastics have a different metabolism. There are biological effects being undertaken, being shown in these cells that indicate differences in metabolism of the cells. And what we really have to do now is try to translate those into a better understanding of the long-term effects of the placenta cells. Again, we have to publish this work, so I can't show you too much of it, but I just wanted to indicate as a take-home message that yes, placenta cells take up different sizes of plastics, and yes, there is a biological change in these cells that indicate that metabolism in the cell is changing. And in order to understand if that's negative or not. We need to do additional studies, and we need to follow the effects for a longer period of time. So these are preliminary effects, preliminary results that really indicate that we really do have to do more research, because these preliminary indications are concerning for us. So to wrap this part of the talk up, yes, microplastics reach the human fetal environment. We have strong indications of uptake in placenta and amniotic fluid, and there is in the meantime a publication confirming uptake in placenta. Can they cause harm to the placenta? Well, we have seen that there are biological responses in placenta cells that require further research. So I can't answer that question yet, but they are concerning to me. And do the chemicals associated with microplastics cause harm? It's too early for me to answer that question as well. So what will we do from here? Well, we will verify that what we're finding in these 30 samples in terms of indications of uptake of microplastics in placentas are really found in larger populations. We will look at the long-term effects of metabolic changes, and we will try to better understand the different toxicity of different sizes and shapes. We will do this within a large new project, which we've called Momentum, which runs from June 2021 to June 2024. And this is a really nice way for us to answer these questions on a broad scale. And I'll just finish off now by introducing this consortium. We are working together with many different research organizations in the Netherlands and many different private sector organizations in order to work together with industry to better understand how micro and nanoplastics are formed, what the internal human exposure to these micro and nanoplastics is, what the hazards are, what the risks are, and together to work on a risk assessment roadmap and to start looking for solutions to these micro and nanoplastic exposures. And while we acknowledge that there are large data gaps that need to be filled, we also acknowledge in this project that we actually don't want to wait until we have all the information that we need to do a comprehensive risk assessment of this form of pollution. We already know that we do not agree, where we do not want our fetuses exposed to micro and nanoplastics, for example. We really want to look at how we can take steps now to have solutions for micro and nanoplastics. So with that, I'll end my seminar.